days, there has been a need for establishing a healthcare system using bioinformation. Biois is working towards maximizing efficiency of the healthcare management system by developing technologies that analyze bioinformation from a drop of blood sample. Our three technologies, reverse SELEX, LPCR, and pCounter, will be a core player that constructs an innovative healthcare management system. Reverse SELEX is a technology that selects biologically meaningful, disease-specific aptamers from a random single-stranded library. First, single-stranded DNA is replicated to double-strand, which undergoes in vitro transcription to construct the random single-stranded RNA library. This library is incubated with diseased patient serum. Ones with higher affinity bind, and then the heat causes the aptamers to be disassociated from the specimen. Now, the aptamer library of the patient is constructed. This patient's aptamer library is used to construct a biochip that can bind aptamers into a glass slide. Then, the set of patient's aptamer library is tagged with Psi3. The other set is incubated with a special reagent that carries proteins of numerous diseases. These selected aptamers are tagged with Psi5. 1-to-1 ratio of the two tagged sets are hybridized onto the already made biochip and put into the scanner for reading. The color of each well indicates the ratio of each aptamer. APTA-CDSS collect this data and categorize the patient into specific stages of the disease. When the database is constructed, disease-specific aptamers are selected and used for diagnosing purposes or identifying novel biomarkers by backtracing. Utilizing the constructed database from reverse SELEX, Biosign is our product line that uses 1,149 of the biologically meaningful aptamers that are specific towards cancers, cardiovascular, and chronic liver diseases. Once the patient sample is diagnosed with our Biosign, APTA-CDSS locates the disease stage of the patient. In addition to Biosign for diagnosis purposes, reverse SELEX is a useful tool to construct databases for bioinformations. LPCR maximizes the advantages of immuno-PCR and simultaneously analyzes quality and quantity of target proteins. There are three ingredients in the E-tube. The first is capture ligand, a conjugation of the magnetic beads to the antigen-specific antibody. The second is labeling ligand, a conjugation of the oligomer to the same kind of antibody. The third is patient sample that carries disease-specific antigen. When all three are incubated, the two ligands bind to the antigen and form a complex. After this complex is harvested using a magnet, we run real-time PCR. There are many advantages using LPCR. First, it can perform multiplexing tests. Second, quality control is easily maintained by comparing the result with the standard target molecule. Third, solution sandwich method allows faster and more precise 3D kinetics. Fourth, it has an excellent limit of detection. And lastly, it has a very broad spectrum of applications. For example, for coronary artery disease, we can determine the ratio of each ligand and find out exactly where the patient is located in the graph to treat the disease accordingly. Same goes for Alzheimer's disease and sepsis. LPCR is not limited to these diseases, but to any diseases that have standard graphs that indicate the ratios of disease-specific ligands. pCounter assay is a digital reading for proteins using aptamer or antibody. The first set of aptamer is attached to the reporter probe, which consists of different colored barcodes with biotin at the end. The second set of aptamer is attached to the magnetic bead. These two sets bind to the target molecule. and only the magnetic bead conjugated molecule is harvested using the magnet. The complex is disassociated and the biotin attaches to the cartridge. When the cartridge gains voltage, the reporters are aligned. And these color-coded reporters are read digitally by the machine to give out the exact number of each protein. The application of pCounter can be anything related to protein-based diseases. The advantages of pCounter assay are 1. Higher accuracy of digital readings. 2. One test multi-targets in the hundreds. 3. Convenience. There is no need for numerous PCR ingredients and procedures. And 4. High reproducibility. With these technologies, we will lead the future bioinformation market as the system biology innovator.